gives me great pleasure today to introduce Jim Kazura, who hopefully needs no introduction. Jim was a former president of the American Society of Tropical Medicine and Hygiene. He was a long, strong time editor of the American Journal of Tropical Medicine and Hygiene. I've known Jim, Tricia and I have known Jim since we worked in Papua New Guinea since the early 1980s, where we first met Jim. A well-recognized authority on both malaria and lymphatic filariasis. So he basically said, what should I talk on today? NTDs or malaria, and decided that malaria might be of greater interest to the group here. Jim is the program director for the International Center of Excellence in Malaria Research for the Southwest Pacific, that's working in Papua New Guinea and the Solomon Islands, including work at JCU. And he's also the director of the Center for Global Health and Disease at the Case Western Reserve University. So, without further ado, I'd like to introduce Jim, and we'll talk to us today about the paradoxes of the road to malaria elimination.
So that you find out Louis can hear you.
Louis, can you hear us?
I can. Hi, Jim. I'm sorry I can't be there in person.
Hi, Louis.
You can hear me? Good.
Yep. Loud and clear.
Okay. Well, thank you, Tom and Trish, for being such nice hosts and hostess for this visit. And I've come through Ken's many times on my way to Papua New Guinea, which I'm going tomorrow morning. But this is actually the first time I've been to James Cook University, and it's really an honor to be asked to present a seminar to the group here. And I've had a chance to talk to some of you here today. And I must say, your campus is wonderful, and it really seems like a very intellectually stimulating environment. It truly makes you feel that you're almost in the tropics, but not in the real tropics quite yet. And I think, to use Kevin Marsh's jokes, this is like being in the tropics, except the power's on all the time. Okay, so well, thank you very much. And so I'm going to talk today about some of the work that we've been doing in East Africa, but try to put it in the general context of attempts to the agenda of eliminating human malaria. And what I'll do during the course of this talk is give you at least my perspective and overview on the impact of current malaria vector interventions in sub-Saharan Africa, with a focus on Western Kenya, and I'll come back to that in a few minutes. I'll then present some data that's been published, but it's really published by a group that's been directed by a Australian colleague, Miles Davenport, who's a modeler based in Sydney. I'm sure many of you work with, and it's been a delight working with Miles for many years. And I'll present some of our published and unpublished data related to using antibodies as biomarkers for protection from symptomatic malaria versus infection with malaria. And then some ongoing studies, which is actually part of an NIH grant application that's under consideration, of what is the impact on transmission reduction, not just simply on serology to malaria, but also malarial disease susceptibility, and that's where the paradoxes comes into the discussion. So, as we know, an agenda for malaria elimination was set in 2007 and 2008 to actually eradicate malaria, with no timeline on this. And I think it's important to say what we mean by eradication. It means like the permanent cessation of transmission in endemic areas. So there's been a lot of discussion about this, but when we talk about elimination, to me that means local eradication, but not geographically worldwide eradication. And this is a map that I think Grant Dorsey prepared a while ago for a publication that we were preparing as a trans-iceberg publication, identifying areas of the world where they're in the process of eliminating malaria and in the process of controlling malaria. So the purpose of today's discussion, I think what's noteworthy is controlling malaria means, to me at least, it means you're controlling malaria at the level that you don't have as much morbidity and deaths attributable to malaria if you weren't controlling malaria. And controlling malaria is simply, as I'll talk about in a few minutes, really is, at least at the present time, is primarily vector interventions as a public health tool. And you can see that where we're controlling malaria is essentially most of the developing world in the tropics. In the green areas are places where malaria used to be endemic, including this part of Australia. It's been eliminated from this continent. Although I must say, it wasn't due to any deliberate effort. It was primarily due to the socioeconomic development. So this is just a pictorial summary of what we have for current public health interventions for the control and elimination of malaria. And this really became a big deal certainly with the malaria Millennium Development Goals that were set in 2000 to achieve by 2015. So the major new things that were introduced, and there was a lot of resource given to increasing the distribution, for example, of insecticidal bed nets. They've gone various iterations. I think some of you may not be aware, but working in Papua New Guinea over the last 25 to 30 years, the actually first bed net trials that were published were in East Sepik Province. There was no insecticide, but there was just the use of indoor insecticides in East Sepik Province. Tom and Trish probably know better than I that when this was done, I think it was done in the late 80s or something like that. So now we have long-lasting and primarily pyrethroid impregnated bed nets. We have indoor residual spraying that is used in many areas, but not all areas. We have rapid diagnostic tests, and what we have now is improved point-of-care treatment with primarily with artemisinin-containing drugs. So that's what the current public health interventions that have been used, particularly in sub-Saharan Africa. So this is from a paper that was published in Nature this past October, as I recollect. So if you look at these data here in the panel A, the more red is the higher the standard epidemiologic measure of level of endemicity, and that can be standardized across different areas. And I'm just talking about Plasmodium falciparum here now. I'm not going to be discussing Plasmodium vivax today. Is the blood smear positive rate in 2 to 10-year-old children. So you can see in this heat map here in panel A that red is 100%. That's the highest level of endemicity. And as it gets more blue, you can see that the lower is the proportion of children in that age group who have a positive or asymptomatic and have a positive blood smear. And you can see simply looking at panel A versus panel B that you see a lot more blue in 2015 than we see in 2000. And this has been consistent with a lot of resources through, for example, in sub-Saharan Africa. It's the President's Malaria Initiative, Rollback Malaria, a lot of other Gates Foundation, other NGO-supported efforts to do that. And if you just go straight to panel D, it looks like the degree of reduction in the PF 2 to 10-year-old group. Look at panel D here. You see in the red, this is the 2000 levels, and this is the levels in 2015. These are data that are collected from generally similar but not identical databases that are maintained either for research purposes or gotten from the ministries of health of these various countries. So that's clearly, there's some confidence intervals that are not expressed here that are important. And if you just focus on panel A in the right-hand side of the slide here, you can see that the major effects were attributable to the distribution of insecticide-treated nets and indoor residual spraying, which much less of an impact related to the use of ACTs in this slide here. So it's been primarily vector interventions that have resulted in this improved malaria control. And we're talking about the malaria burden here. This is symptomatic infections with uncomplicated malaria. There are not comparable data, to my knowledge, looking at severe malaria, which is a much more difficult diagnosis to make from a research perspective. But there tends to be a downward trend in severe malaria, which could be coincidental with the use of artemisinin drugs for treatment of severe malaria, and its recommendation that it replace quinine or quinine for treatment of severe malaria. So that's kind of the good news. Those are all appear to be very positive things in terms of implementation of vector control. So this is a grant that, I'll go through the history of this in a minute, but this is a grant I applied for in 2009 to 2010 for competing renewal. So I've been working in Western Kenya near the Kisumu area. This is in the same labs where I think some of you may have worked before. They have Walter Reed and the CDC have laboratories there. It's with the Kenya Medical Research Institute. So I applied for the third competing renewal of this grant, which started in the early 90s. And it was not actually, it was triaged the first time through, because when we wrote the grant, the papers had just come out from Kalifi, from the Oxford group and Kemry that are working there, that they said he can't study malaria there because the malaria is gone. Okay? And so this just shows you they have, I mean, the one thing is that malaria, just like other infectious diseases that we study in the real world, often are very heterogeneous in their distribution. So I didn't know where they were coming from in this thing, because so we actually went through explaining the epidemiology in this area, but the reviewers just glummed on some of these recent high-profile papers showing that malaria was, and uncomplicated malaria was disappearing from coastal Kenya, which we observed at that time as well in the south of Mombasa, as well as the Wellcome Trust people did north of Mombasa in Kilifi. So the area that we work in here is in Western Kenya. You can see Kisumu is kind of the largest town in this area, and it's all around this Sembo Bay area that we're doing this study. So our first phase of this is we studied pre-erythrocyte and blood stage immunity. It's interesting to see that these grants titled on track along what the research topics were at the time, because people were much more at that time interested in pre-erythrocytic immunity. We had a grant to study that about for 10 years, along with, to a lesser extent, blood stage immunity, comparing lowland areas here to more highland areas where there was much less malaria, such as near Eldoret. The second was focused on MSP1, and we had like 7 or 8 years funding. This is work that Brendan Crabb was involved in the early days of this with some of the earlier transgenic parasites that were made to look at functional antibodies that targeted the C terminal region of MSP1. That grant, we know was not going to go anywhere, because the earlier vaccine trials of the single allele and the wrong one that was tested for MSP1 in Western Kenya showed no efficacy. I thought that was premature, but that's the way the world works. So MSP1 was kind of, at the time, was off the table for a blood stage vaccine for Plasmodium falciparum. So then we go up to the current time. So things have really changed in Kenya here. I mean, first of all, healthcare has really improved. This is a picture of a Luo ethnicity person. Some of you may recognize this face of this man. This is Barack Obama at his father's home. There's one of him smoking a cigarette. Okay, but I thought that was politically incorrect to show the President of the United States smoking a cig.
He wouldn't have been held.
This is when he was an alderman still in Chicago. He wasn't even, this is before his days in the Senate. And I think this is his grandmother. This is his father's mother here. Okay. Interestingly, he has not gone back to Luo area. Part of this, even when he was in Nairobi a few months ago, he deliberately did not go there because there's tension between Luo and the current government in terms, which is a long-standing kind of thing. So it's kind of a fun part of it. Parenthetically, the very first time I ever went to Kenya was in 1974. I worked with some people there when I was a medical house officer. But I worked in this exact same area of Kenya on schistosomiasis in that area, which is where Dan Colley and those people just work. And there's still a hell of a lot of schistosomiasis there. So these diseases, at least there's less illness from it, but there's still a lot of infections there, which I think is the challenge for some of these elimination and eradication agendas. So, okay, so this is kind of, and I'm not a vector biologist and I'm not an entomologist, but I'm aware of its significance to looking at immunity and pathogenesis of disease in humans who live in these malaria endemic areas. And this is some work that's taken from some of Ned Walker's work that's been done off and on over the last 20 to 25 years in Western Kenya. And it shows the proportions of Anopheles gambiae and funestus that have have changed, but you can see with vector control here, you have the reemergence of Anopheles funestus, which actually historically has been one of the major vectors, but reappeared at this site, coincidental with improving vector control. So when you say improving vector control, I'm primarily talking about the distribution of long-lasting insecticidal nets. And I must say, we personally do not have data from the identical villages that we work in now in terms of how much the bed nets are used. Okay, so this is kind of like loose association kinds of things. But the Ministry of Health in Kenya got serious about distributing insecticide nets in the area we work in 2005, 2006. Some of these changes occurred coincidental with this. And this is from some of their work here. We can see that although Gambia still has the highest infection and CSP positive rates, it's not numerically the most common vector. And again, this is not my area, but there is a shifting of the peak biting time of these vectors to earlier and earlier in the daytime than was historically the case. Again, this is work that NET has done. So this has occurred, perhaps predictably, under the pressure of indoor residual of the types of bed nets that are being used there. So if we take this standard measure and we look at our own data from asymptomatic children that are 2 to 10 years old who were at that age in 1996 from the same area that we work in now in 2003 and 2013, you can see a remarkable decrease from about 80 to 90% to 18%. If we'd done PCR, which we didn't do in 1996, but we had the samples from 2003 and 2013, by PCR for Plasmodium falciparum, it's 100% in 2003, and it's about 40% by PCR in 2013. So there's been a significant reduction in that, however you want to use that kind of metric. And this is unpublished data, but our study is focused on uncomplicated malaria. This is kids who come in that are 10 and under, who are RDT positive, have a history of fever, have documented fever, and by the judgment of the local clinicians, are thought to have uncomplicated malaria, and are given a 3-day course of co-Artin. And just to show you, this is like between 2012 and 2013. I'm not going to talk about data today. We're doing about these data. We're doing studies on B-cell immunity in these kids, as well as in monocyte function in these kids. We get a sample when the kid presents with acute malaria, and then we get a convalescent sample 6 weeks later. And we only aimed at getting about 200 kids enrolled in this. We could have easily enrolled over 500 to 1,000 kids per year that meet our strict criteria for uncomplicated malaria. So there's certainly, this is data from the single clinic where we see children that get enrolled in that study. This is data from the CDC KEMRI demographic surveillance system that shows you the trends in malaria mortality in the local teaching hospital, which is where all the kids would. So if we see a kid that comes in our clinic, which is about 15 kilometers from, which is a place called Chilango, that child, if in the clinician's determination that kid is thought to have severe malaria, have seizures or something like that, they are then transferred to this teaching hospital where they have much better laboratory facilities. So these are real malaria. So this is malaria attributable mortality in kids who come in this area. You can see commencing in about 2008, 2009, an increased malaria attributable mortality in 5 to 14-year-old kids, and as well as kids under 5 years of age. Okay, we haven't studied these specific individuals, but there's a suggestion that there's increasing severe malaria cases that are now emerging, again coincidental with the increasing distribution and reducing malaria exposure, but still with continuing transmission. And in retrospect, some of these things might have been predicted by the early days of using mass treatment with chloroquine, for example, in the WHO efforts to eliminate malaria, which involved using chloroquine this mass drug administration. Okay, so the first thing I'm going to talk about is what do we talk about when we're talking about naturally acquired immunity? And so we're trying to get some in vivo insights in what this means to people living in malaria endemic areas. So this is a study that we did in 2003. This was done by someone who was a junior faculty with me at the time, Ann Mormon, who's now at the University of Massachusetts. And what we did is we did a treatment time to infection study. And actually, Alex and I were talked about this earlier about how you look at this in endemic populations. So what we did, we went to a village and we looked at 200 healthy people from the ages of 1 to over the age of 18 years of age. Okay, these were all asymptomatic individuals. We looked at malaria transmission in this part of Kenya is perennial, but there's seasonal variation. Okay, so before the long rains come, when we knew we anticipated there would be more transmission, we gave everybody informed consent and gave them a 3-day course of Coartem, so to speak, clear them of infection. We then looked at those individuals 2 weeks, and this was under direct observation treatment. We then did a blood smear at 2 weeks, and if they were blood smear negative and they were PCR negative, we assumed that they were cured of malaria. So they were like cleared of the infection because the Coartem wouldn't cover liver stage falciparum that might have been there when we treated the individual. So if they were positive, we assumed we didn't consider them for entry into the study. We then got a venous blood draw on these individuals at the beginning of this period of time, and then got weekly blood smears on these individuals and did active surveillance for clinical malaria. So what we could do then we looked at blood smear changes over time, and we looked for, if an individual had symptomatic malaria, we documented that, and then we gave them another dose of Coartem. So what we did then is we compared PCR positivity, which is in the lower part of this here, to blood smear positivity, and then we took a week before and after that as the apparent in vivo parasite growth rate. Now, caveats in this: these are certainly not. These are probably multiclonal infections of what you expect. In fact, we know that. But it gives you an average parasite growth rate that is in vivo. And we compared that for individuals at according to age. And here are the raw data. So if you just look at the PCR data, which is the solid line here in the first panel, and here you can see this is 1 to 4-year-old kids who, when they become PCR positive, and you look at the blue dotted line for that same group, you can see there's a relatively short period of time. For those of you who can see the screen, it's a relatively short period of time between the time, on average, people became PCR positive and became blood smear positive. Okay, some of these people who were blood smear positive were sick, and some were not. Okay, so this is really in vivo. Includes both those groups here. So if you can contrast that with, say, people who are 18 and older, we excluded individuals between the ages of 15 and 18 because of some complex issues related to puberty and gender at that time in terms of malaria immunity. And you can see here, blood smear positive occurred much later, PCR positive in adults, and a much more delayed time. And this is expressing it in terms of the actual days on the right-hand side of this panel. If you then convert this to the parasite multiplication rate, you can see that on average there's a higher parasite multiplication rate for the youngest children, 1 to 4 and 5 to 9, relative to older children as well as adults. And there clearly are outliers in both groups of people. So this is kind of the in vivo measure of actually what naturally acquired immunity, or one aspects of age-dependent naturally acquired immunity looks like in 2003. We cannot do this study today because at the time we did this study, you could do blood smears on someone and look at it in Kenya, and if they were negative, you did not treat them. That was the standard of care in Kenya. Correctly or incorrectly, the official policy for the Ministry of Health in Kenya, which isn't true of all sub-Saharan African countries, if you do a blood smear on someone and you look at it, then you must treat them whether they're symptomatic or not. And this is a variable policy in different parts of Africa. It's an interesting discussion on what that really means. Some people think that you may actually extinguish the development of immunity. I don't know what the answer to that is. And you certainly can't do a controlled clinical trial to answer that question in many parts of sub-Saharan Africa. Okay, so the next part of the talk will be really looking at the antibodies, and this is work that we've done with Bill Belgner, and I know that many of you have worked with Bill, and this is looking at the use of these Plasmodium falciparum protein microarrays, taking genome sequences and looking at the proteome at the same time, and then developing, recombinant proteins of about 600 to 800 amino acids each, putting them on a chip and looking at antibody activity against a whole broad array of known as well as poorly annotated open reading frames from these parasites. And this has been done for a lot of this, and it's an obvious limitation of this. First of all, in the case of Plasmodium falciparum at the present time, it's related only to the 3D7 sequence, the genome sequence, and the 3D7 proteome, which has been maintained in the laboratory for over 30 years now. So it's unclear what this means in terms of the genomes of a lot of real parasites that are infecting people in sub-Saharan Africa at the present time. So this is a heat map here, and you can see the red is stronger antibiotic activity. This is 86 adults in the x-axis, and the y-axis. This is the 163 PF reactive protein features, if you will, or antigens that are in this array. And there's something like this chip had about 1,700 different proteins. So again, the vast majority, there's no reactivity. This just showed. And you can just look at this. You can see adults have a stronger reactivity to a broader array of proteins than to the youngest children, and it tends to increase with age. And the right-hand part of the thing shows, of this heat map shows that minimal reactivity with malaria naive human beings. This is taking the 163 most reactive antigens based on adults. We figured those have the greatest exposure, those are the most immunogenic, and the adults would assume they have the greatest cumulative malaria exposure. And you look at the signal intensity to these different proteins from 1 through 163. You can see that the children who we observed over the first of all, looking at time to reinfection and looking at these data, there is no significant relationship. And we've used a lot of fairly unbiased statistical tests to look at that. So there is no relationship between the patterns and strength of antibiotic activity that are present in the plasma of these people before, and some people got reinfected and some people did not get reinfected, or the duration of time to infection, there was no significant relationship. Then we looked at it in terms of symptomatic malaria. And the only people who got symptomatic during the course of this observational study were individuals who were children, okay? And we've adjusted for ageness. So we compared children who were unprotected compared to children who were protected. And you can see that the children who were protected had significantly greater reactivity than children who were unprotected. And this just shows, this corrects for multiple comparisons. The longer this downward lines are, the higher the significance of the relationship to protection from symptomatic malaria. If you compare that to children who were protected to adults who were protected, you can see just looking at this that there was a greater similarity in reactivity between kids who were protected versus adults who did not get symptomatic malaria. So these are a bunch of this is ranking from the most significant to the least significant, and you can see some of the usual suspects in here that people have been talked about from vaccine perspective. MSP2, LSA3 was actually something that was described by Pierre Driel in chimpanzees that's expressed in the liver stage as well as the blood stage. And you can see there's a whole bunch of PFEMP1, different features of the 3D7 sequence. I might add, and we don't have time to talk about it, but all the PFEMP1s that we see in here were all from the intracellular, highly conserved domain of PFEMP1. To the highly variable head structure, there was no significant relationship. And that head structure is just, again, based on the 3D7 sequence for all 63 or 60 or 63 bars that are present in the 3D7 sequence. Okay, so now going to the issue of what impact does the vector control have on potential malaria rebound, if you will, and to naturally acquired immunity, is we then started looking at these the antibodies, serologically at least, to the PFEP1s among asymptomatic children and adults. So this is just looking at pre-erythrocytic proteins, first of all. Okay, so you can see here, this is, if you go from right to left here, the youngest individuals are 1 to 3, then 4 to 6, and 7 to 10, then adults. And you can see that, for example, with PFCS, this is two different constructs of CSP that we use, LSA1 and PF celtose. This is for just focusing on this construct of this is a full-length valcircum CSP1 protein. You can see there's an age-related increase in reactivity for CSP. You see that trend in 2013 for age-matched children in the same group, but you can see that there's much lower increased change with age in this group, and on average there's a significant lower antibody titers consistent with decreased exposure to sporozoites in this population that is age-matched for 2003 versus 2013. We won't go through this whole bloody slide, but this just to show for a selection. We've done this for other blood stage antigens. You see the same thing for MSP1 3D7. I like this one here because this is a very interesting vaccine candidate. Here's MSP3, which a lot of studies have been done on, and we can see the same thing here. But again, the levels of antibodies are significantly lower for MSP3 in 2013 versus 2003. Now, getting to the virulence protein issue, is we're also focused on the PFEMP1. As many of you are likely aware, PFEMP1, there's a certain structure of the domain cassette 8 and 13 that this region, the CID or R1 alpha region of PFEMP1, binds the endothelial protein C receptor, and that particular interaction is thought to have cause a lot of vascular dysfunction and account for severe malaria to a certain extent, particularly in cerebral malaria. So using some of these proteins that Smith has provided for us, which we believe are correctly folded, they actually bind it to endothelial protein C receptor. Just focusing on that, this is what I just said, I'm going to skip this here. We can see this is the changes in the antibodies, the CRD alpha domain, that's 8 and 13, these are the ones that bind to EPCR. You can see here, here's 100% of kids by the age of 3 in 2003 had antibody responses to it, where it was 70% in domain cassette 8. So it's decreased in this time, and you can see that there's much lower levels of these antibodies to domain cassette 8 as well as to domain cassette 13 in 2013 compared to what we saw in 2003. There is currently no functional assay to show that these antibodies actually function to prevent severe malaria. This is only serologic evidence for such. So I guess what I'd like to conclude with is saying that continuing vector interventions are appropriate and will likely affect malaria epidemiology and natural immunity to uncomplicated and severe malaria. And it takes something that Tom and I have talked a lot about the utility of indoor bed nets, for example, in the Solomon Islands, where the mosquitoes bite outdoors. It does protect you against the mosquitoes that bite indoors. There just may not be very many of them. So, but clearly we have to, in my view, we have to accelerate the development of vaccines that will mitigate malaria pathogenesis of this disease, as well as those that will facilitate transmission reduction. I think, from my personal perspective, we need to reinvigorate the field to develop blood stage vaccines that can prevent disease. I think we're going to be seeing more of this malaria resurgence the more successful we are with really reducing malaria transmission, but not stopping malaria transmission. Current approaches to eliminating malaria in high transmission areas in some areas of Africa are not likely to succeed. I guess that's a politically correct way of saying it. There's a paper that came out from Tun Busama and others from the Highlands in Kenya this last week in PLOS Medicine, where they gave MDA with intense vector control in a highland area to hotspots that they identified in a relatively low hypoendemic area, but there was no impact on CS positive rates of mosquitoes except for the first 6 weeks, but by 3 months later, there was no difference between areas that got the MDA plus the intense vector control plus versus those that got the standard of malaria control in Kenya. And what I don't have time to talk about today, but I'm happy to talk about those who are interested in it, I think it is possible to accelerate the development of natural. Is it possible to accelerate the development of natural immunity to malaria by targeting inflammatory pathways associated with malaria pathogenesis? I believe it is possible to do that, but we need to get much more basic information about why immunity to malaria is so slow to develop in endemic populations. What leads to this inflation of these so-called atypical memory B cells? And how does inflammation play into that? And how reversible is that? So those are the kind of typical questions that I think would warrant research in the foreseeable future. And thank you for your attention.
Okay, we'll throw it out to questions, and as always, we'll throw it out to Townsville first, since at some point in time the video will go off. Do we have any questions from Townsville?
Hi, it's Louis. I hope you can hear me. So that was really lots of really beautiful stuff. Jim, I just wanted to ask you particularly about the sort of the B cell story that you alluded to at the end, and essentially this is a kind of failure of B cell memory. So I just wondered if you might choose to tell us a bit more about your thinking and what might be going on there in terms of current stuff. I've been a little out of contact with current thinking. I'd really appreciate a kind of update or a brief.
Okay, so
it's all right.
Okay, what I'm going to show you is unpublished observations. So first of all, one thing we've noticed with our B cell studies, so these are kids who come in, these are all kids like, in this is 9 1 to 5-year-old kids presented with acute symptomatic malaria. Okay, so we get the blood before and then the kid is given coartem. So we take those cells back to the lab, we cryopreserve them, we get plasma, blah, blah, blah. Then we go back 6 weeks later, and we document that the kid is cured of malaria. The kid is blood smear negative, and he or she is healthy. Don't have time to show the data. Within a day or two, we go to that same village where that kid came from, and we get a kid who's age mapped switch in a year, who's asymptomatic, and we get blood from that kid. So we have a simultaneous control. Okay. So when we attempt to do B cell ELISpot, so for the non-aficionados, this is a standard assay. What you do is you take the peripheral blood mononuclear cells from these individuals, you do a polyclonal stimulation for, say, 4 to 5 days with a cocktail of CPG, et cetera, to lead to transfer. So you get the memory B cells expanding, and then you take those cells and put them on a plate to do an ELISpot to look at antibodies produced to MSP1 or MSP3. So when we do this is so we also know the number of IgG secreting memory B cells. These are not necessarily malaria specific. They represent all the B cells that can be polyclonally stimulated. So these data show that, first of all, we cannot even measure tetanus specific memory B cells very well in these kids. This is the polyclonal stimulation. These are from the same individuals here. You can see this is the antibody secreting cells of 5,000 cells from these kids. This is polyclonal stimulation. There is a direct suppression of even the memory B cell production of IgG and during an acute malarian attack. Okay, but then it recovers significantly at 6 weeks. Okay, and we've looked at these kids. Some may actually be PCR positive, but but there's no relationship to people. But they have the same level of polyclonal stimulation. So first of all, we cannot do valid memory B cell ELISpots with B cells from kids presenting with acute malaria. Okay? We attempted to look at this by doing it ex vivo by using Q dots and stuff. But again, these are 2 and 3-year-old kids. So we get 2 milliliters of blood from these kids. So first of all, and we've actually looked at the colonies forming from these kids, and you can see after 2 or 3 days, you don't see colonies, you don't see any of these colonies forming that you see where you're getting memory B cell development. So this is a black box as far as I'm concerned, but we're very interested in this. It correlates, and here's looking at the different memory B cell compartments, okay? So here we can see this is the classical memory B cells. These are CD19 positive, CD27 positive, CD128 positive memory B cells. And there's a slight difference here, but this is acute malaria versus 6 weeks. There's not much, it's a slight difference, but it's really attributable to 2 or 3 kids here. But you can see these kids here are in the same range. These are healthy children from the same village, and here's healthy adults from Kenya. We don't have it here. If you look at healthy adults that are in this room, they have many fewer memory B cells in their peripheral blood simply because people they have schisto infections, they have all these, they're much more experienced microbial infection-wise. So the memory B cells, the actual numbers of that we can detect by flow cytometry are not depressed. Here's activated memory B cells, and this is no surprise here. So again, we see here, there's really no difference, again, between acute malaria in 6 weeks. Here's healthy kids and healthy adults again. But what we see here is this remarkable inflation. And, you know, Peter Crompton and Greta Weiss published now in 2008 this expanded atypical memory B cells in kids from Mali. They were asymptomatic but living in a malaria endemic area, and there was greater proportions with increasing age. This is what we see here is a remarkable expansion. Some of these kids had up to 60% of their B cells at the time of presentation of acute malaria were atypical memory B cells. Okay, so so what's the intrinsic defect here? Is it an intrinsic B cell defect? Is it a defect in T helper cells, as T follicular helper cells, which is another possibility. So this is one of the things that when I talked about, if we can determine what this is, this may give us some insights even, and let me, if I go back to a slide here, my personal view is, this is looking at plasma, this is ex vivo cytokine levels on these same kids that I just showed you here, okay? And what you can see, there's acute malaria, 6-week recovery. IL-6 is off the wall here. Acute malaria goes down. Here's BAFF, which is a B cell development cytokine, okay, is elevated at acute malaria. TNF alpha is markedly elevated. IL-10, IP-10, which is which has impact on T cell activation and B cell activation, and interferon gamma. So I think it's a lot of this is systemic inflammation. We're now stratifying this for PF-HRP2 levels because when you do a human study, they come in with acute malaria. Some kids have been sick for a day or two. Some kids have been sick for 6 hours. So different amounts of counter-regulation have already occurred. But I think these are the kind of issues where the human studies with an experimental challenge are helpful, but this is where doing this kind of thing in the field will really be challenging. But we need to be able to do studies of sufficient power where we can account for some of this expected heterogeneity. That's a long-winded answer. So I don't have answers to your question, Louis, but I have some.
Look, it was in general. I thought that was really interesting. I think there's a lot of meat to be teased out there, and you've got some, particularly with the atypical B cells, you've got something that really deserves more biology to bring it up and to expand. And the general failure in polyclonal activations, I think it's very interesting.
Am I remembering correctly? There was a paper a while ago from Michelle Wax and Michael Good that talked about increased apoptosis of B cells. Was it B cells? They talked about increased apoptosis.
It was with dendritic cells, I thought.
DCs.
And that was primarily myeloid dendritic cells because, I could be wrong about this, Alex, but I think in humans, plasmacytoid dendritic cells, which are the ones that you're mainly interested in for antigen presentation, and they produce like a lot of type 1 interferon and things like that. Doing functional studies on them is very challenging in humans because they do not survive cryopreservation very well. Unlike myeloid dendritic cells. I think we're talking about this. It was from Papua New Guinea, I think, where they got some of their PBMCs.
Yeah.
Yes, I've talked with Michelle about that, yeah. I try to stay away from the immunology stuff because it's a somewhat arcane area. But
Anybody else in town?
Yeah, anyone else in town have a question? Janice?
Now, mosquito question.
yes, okay.
How much of an issue is pesticide resistance?
From agricultural pesticides?
No. In for malaria, for your anopheles in Africa.
Oh, well,
With
there's two people in the room that know about 10 million times more than I do, but Tom and Tricia. But I think that in Western Kenya, I think that there's pyrethroid resistance. I think, and actually we're applying for an isomer where there are people that are trying different kinds of insecticides. Look at their level of. So I think that pyrethroid resistance is, especially like places like Ethiopia, is for most anophelines that transmit malaria is 80 to 100%. I don't know. I don't know the numbers, but
Yeah.
Tom, yeah. So it's all.
It's high.
That preceded even the bed nets in Ethiopia. Where some of this was indoor residual spraying or the use of DDT in the past. And what's remarkable is we don't see this in the South Pacific, which I don't quite understand why we don't see that. But,
We are seeing a little bit of resistance of pyrethroids, and for the first time in Solomon Islands, Hugo Bogor has shown some moderate levels of resistance. And that's a recent development. It's one of the conundrums of malaria control is that despite widespread resistance to pyrethroids, which is the only insecticide we can use on insecticide-treated nets, there's really no evidence of programmatic failure or reduction in efficacy of the net. Now, whether that's a function part of just a protection, physical protection, that barrier that the net provides or not. But it's one of those things, it stands to reason at some point in time that you will see impacts on efficacy of the nets. And the classic place of that is really Ethiopia. When you look at this great DDT, they had a reformulation plant in Ethiopia, and they sprayed DDT for about 50 years. And when you actually did insecticide-resistant testing, something like 99% of mosquitoes were resistant. You could go into a house in the morning and collect plenty of Arabiensis resting on the DDT sprayed walls. They did their all night, very pleasant, very comfortable environment, Thank you. But it will be, it is a cause for concern, but I think the message to the country programmatically is that don't stop doing what you're doing. As the data that Jim showed in that paper in Nature attributes 68% of the reduction in malaria due to vector control. So it's still working.
yes.
We should be concerned, but don't stop doing it just because you're seeing resistance. There's a lot of concern about trying to integrate resistance management plans in the countries right now and do insecticide rotation. But we have the problem with bed nets, on which most of the control relies, and pyrethroids are the only thing that are safe enough to be recommended in the present configuration of the bed net.
Jim, the proteome array work. Now, I think all the data you presented was just polyclonal IgG responses, right? You didn't look at subclass-specific responses?
No, I always want to do it, but the people I work with said, no, not again. I mean, how many people have done it? Okay, so it's IgG1 or IgG3. Okay.
Do you see IgG4 in a malaria infection, particularly
And that's the shortest half-life of any of them. So James and I are doing some stuff together and stuff, and I think the IgG1 and IgG, looking at functional stuff, I think it's really important because they have different affinities for FC receptors. I think we have to read, but people that got bored with doing this so much because it's always IgG1 and IgG3, both of which are complement activating, subclasses of IgG. But I must say, we've done this in the past, and I think others have. It's uncommon to see IgG2 and IgG4 uncommon. There's old literature on IgE in this. Maria Troy Bloomberg has done a lot of this stuff 25 years ago now, stuff like that. Where are you going with that?
Well, where I was going, in two directions. One was just with the worms and the proteomerase, we get very, very different.
IgG4 is great with worms.
And even 1 and 3, we get very different antigens recognised and the intensities of the recognition difference. So that's clearly not happening with Plasmodium. But the data you showed, were they mono-infections or were some of those kids infected with helminths?
We don't know. So we don't, the only way we know that is by getting a stool sample or doing, we don't have that information.
Yeah, I'd love to know.
That comes up, like actually I'm reviewing a paper now for Malaria Journal. And I don't think anybody in this room is on it, but it's one of the usual ones. What's the impact of Mercator americanus infection on malaria morbidity? But, I'm not saying it doesn't have an impact, but it's not going to account for 30% of the variants, okay? Because, so I think all these things do have an impact on pathogenesis. Infection susceptibility is a different kind of question, but I think most studies are seriously underpowered to do that. And it depends on the kind of, but like with these kids with these different cytokines I showed you, this would be useful for us to understand some of that variation, just to look at potential confounders that are biologically plausible. But it won't quantitatively answer the question. But I agree that I think that certainly, and there's papers out there showing that helminth infection mitigates severe malaria. But you can find yes and no answers to that depending on where you look. But I think they're all sufficient, they're fairly underpowered. I don't know what your sense is about that. I think it's a challenging thing to do because you work with humans. You're asked for poop. And actually in Papua New Guinea, we have a much easier time getting blood than getting fecal material. Much more difficult. There's much more, cultural. It's understandable, to be honest with you.
And you can't be sure, when they deliver a fecal sample, you can't be sure whose feces is actually being delivered.
So, to me, the most interesting slide was the one with the change in the proportional mortality going into the older age groups.
Yeah.
But it wasn't absolute mortality, right? So the absolute mortality
was malaria mortality. Yeah.
It was malaria. It was the proportion of mortality that was related to malaria. And do you think, so, I mean, the implications of that are, that's it, you went past it. So malaria mortality is a proportion of all-cause mortality. So it's actually going up in absolute
proportion
terms.
Proportion of all in that age group admitted who died with, I think I'm reading that correctly.
So, I mean, all-cause mortality could be going down, but malaria mortality is
right. Yeah, absolutely true.
But, I mean, so the implications of that are, sort of the opposite to Rumsfeld thing. Rumsfeld always said, don't go to war unless you're going to blast everything out of the way.
Shock and awe.
Shock and awe. So, I mean, so if you do that and you eliminate, that's fine. But if you can't do that, it's better not to go all out, but to do it slowly. Is that what that implies?
Well, I'll take a middle road on that because I think it's the only realistic road. When I said a blood stage vaccine, I know we've been here before, okay? But these are real-life issues. We don't have a way of eradicating malaria at the present time. So we have to anticipate that, let's see, I don't know, what is your belief about RTSS? It did decrease morbidity in a certain age group for a certain period of time. There's a suggestion that there may be increased severe malaria. Just a suggestion. Includes about the RTSS vaccine in the high transmission areas, such as in Siaya, in Kambewa, in Western Kenya, which is right juxtaposed where we're working. Does it mean we shouldn't use the vaccine? Well, it means the vaccine's not very good, but it doesn't mean we shouldn't use it, because this can be anticipated, even though we don't understand what are the critical factors in losing immunity with decreased exposure might be. It doesn't mean we shouldn't use things that can prevent things now, here and now, but we should anticipate finding ways until we get to a point where we can release a mosquito that's refractory and drive these susceptible mosquitoes out of the population, which will not happen in my lifetime in sub-Saharan Africa, I don't believe. And rekindle some of the more intermediate steps